I've always been interested in understanding of the pathomechanism of different diseases, how genomic variations may cause different diseases, how these alterations can be used to target, cure, predict a disease. So this has always been a drive in my career. My name is Csaba Bodor. I'm a professor in molecular oncohematology, and I work at the Department of Molecular Pathology within the Institution of Pathology and Experimental Cancer Research at the Semmelweis University in Budapest, Hungary. Our laboratory acts as a national center for oncohematology, focusing on acute leukemias, myelodysplastic syndromes, multiple myeloma, aggressive lymphomas. Our laboratory is providing service for more than 20 different clinics across the country. We would like to understand the pathomechanisms leading to development of different lymphomas and leukemias, and also we aim to understand mechanisms associated with progression and resistance to targeted therapies in hematological malignancies. We have a plethora of methods, starting from morphology, including immunohistochemistry, flow cytometry, conventional cytogenetics, FISH, as well as next-generation sequencing is part of our portfolio. And recently, we are actually witnessing the revolution of cytogenetics with the advent of optical genome mapping, which is becoming part of the everyday routine for a more precise, advanced mapping of karyotypic alterations in hematological malignancies. One of the challenges in traditional cytogenetic methods is the failure rate and the lack of dividing cells, the time-consuming workflow, and the need of extensive cytogenetic expertise. There is a pressure for a fast turnaround time when talking about acute leukemias, especially in the pediatric setting. In hematological malignancy, structural alterations affecting larger chromosomal regions are becoming increasingly important. Optical genome mapping is providing a solution for even these cases with a much higher success rate. With optical genome mapping, we don't need dividing cells. The workflow is much faster, smoother, and the analysis is automated. So there is no need for a long-term cytogenetics training, but the software is able to help to generate karyotypes in a very smooth way. Our laboratory is running the National Pediatric Leukemia Molecular Profiling Program, and optical genome mapping will become a standard assay within this program in addition to the different next-generation assays being run. So practically we are moving from cytogenetics to digital cytogenomics. The workflow is much faster, much more cost-efficient, and the results are actually bringing data on a much more comprehensive level.